Here are the materials that we need in isolation of DNA using a banana. Here are the procedures.
Dissolve the table salt and liquid detergent in 1/2 cup lukewarm water. Stir and mix well. Peel and cut the banana into small chunks. Place the banana chunks inside a blender, then add 1/2 of water and 5 tablespoons of the lysis solution. Blend at the medium speed for 30 to 45 seconds.
Filtrate the mixture over 2 layers of cheesecloth on the top of the strainer. Collect the filtrate into a bowl. Transfer the filtrate into the clear glass. Allow to settle for 2 to 3 minutes.
Slowly and carefully add the ice-cold isopropyl alcohol onto the filtrate. Do not stir the mixture. Observe the formation of white fibrous mass on the interface of the two liquids, which is your precipitated DNA. The lysis or disintegration of tissue or cells is frequently the first step in isolation of DNA. This process is required for the breakdown of protein structures and the release of nucleic acids from the nucleus. Lysis is performed in a salt solution containing detergents to denature proteins or proteases or enzymes that digest proteins. Cells are broken down and membranes dissolve as a result.
A detergent or dishwashing liquid is used to cleanse dishes by eliminating fats and proteins. In the DNA extraction technique, it works in the same way, separating the lipids and proteins that make up the membrane that surrounds the cell and nucleus. The DNA is released from the cell once this membrane have been broken apart.
The purpose of the table salt in the isolation of DNA is that the salt balances the negative charge on the DNA strand, allowing them to bind together. By adding salt, proteins and carbs precipitate as a result. By this, we can help neutralize the DNA charge and make the molecule less hydrophilic, which means it less soluble in water. So the salt also aids in the removal of proteins attached to DNA and keeps them dissolved in water.
Except for the DNA, everything dissolves in alcohol. The ice-cold isopropyl alcohol draws water from the DNA molecules, causing it to precipitate or solidify and appear. Then the DNA will appear as white mucus stands.